Hi, everybody, and welcome to another Fragile X research seminar sponsored by FRAXA Research Foundation. We have been conducting these very popular seminars in an effort to make up for the fact that we haven't been having any in-person meetings lately. We think it's really important for researchers to get together and present their results. And so we're attempting to do that in a virtual format. Just tell you a little bit about FRAXA and the work that we do. FRAXA Research Foundation is a global leader in funding Fragile X research. And we are looking for a cure for Fragile X and along the way developing innovative new treatments for Fragile X. We're really proud that July 22nd will be World Fragile X Day, and FRAXA is attempting to bring together the Fragile X community all around the world. We'll have well over 200 landmarks and monuments illuminated in FRAXA teal, as well as some of the local national colors in honor of Fragile X research and awareness of Fragile X syndrome. But I'd like to introduce today's speaker, who's Dr. Xinyu Zhao from the University of Wisconsin. She's the Jenny and Kyle Professor at the Waisman Center in the Department of Neuroscience. Dr. Zhao graduated from Peking University with a bachelor's degree in biology and earned her PhD in pharmacology from the University of Washington. She then obtained postdoctoral training from Dr. Fred Gage at the Salk Institute. And Dr. Zhao started her own independent research at the University of New Mexico School of Medicine in 2003, then moved to the University of Wisconsin in Madison in 2011. She is currently a Jenny and Kyle Professor in the Department of Neuroscience, an investigator at the Waisman Center, and her research focuses on understanding the molecular mechanisms that regulate brain development with the goal of developing novel treatment strategies for neurodevelopmental disorders. So she's our kind of scientist. During the past 15 years, Dr. Zhao's group has used both mouse and human neural stem cells to interrogate the functions of FMRP in brain development. They've discovered that a lack of FMRP leads to altered neural stem cell self-renewal, neural differentiation, and neuronal maturation. They've shown that FMRP deficiency results in mitochondrial deficits, and using a combination of CRISPR gene editing, genome-wide target identification transcriptomics, cellular imaging, and behavioral techniques, they've identified novel molecular and signaling pathways underlying these deficits in both mouse and human models, and discovered potential treatment targets for Fragile X syndrome. So with that, I'd like to introduce Dr. Xinyu Zhao and her presentation, Interrogating the Functions of FMRP in Brain Development Using Stem Cells. Xinyu?
Hi, everyone, and good morning. And first I want to thank Mike, Katie, and FRAXA for the invitation, for giving me the opportunity to talk to the Fragile X community, both scientists and families and physicians. And so let me first share my screen. Okay, so today I'm going to talk about the work we have been doing for the past 15 years on Fragile X using stem cell as a model. And first, I would like to declare that I'm serving as a consultant for design therapeutics in San Diego. So my lab is interested in how gene regulate development and how that impact behavior and the disease state. And in addition, we're also interested in how genetic disease and behavior deficits can impact development and in turn affect gene regulation. So we use both the stem cells and the mouse models and as in our studies, and we investigate how genes are regulating both systems in vitro and in vivo. So one of the genes that we have been studying for, as Mike said, over 15 years is this gene FMR1, which codes for the Fragile X mental retardation protein FMRP, because the loss of this gene function leads to Fragile X syndrome. And when I became interested in Fragile X over 15 years ago, and the reason is because this learning deficit present in the Fragile X patients. The reason of why I'm interested in the learning deficit is because I was working on hippocampal function, and in the context of a brain plasticity, learning, memory, cognition, emotion, as well as adaptation. So hippocampus is a brain region that both in human and in rodent brains mediate in this lifelong brain plasticity. And especially in the adult brains, this is important for learning, memory, cognition. And one of the unique features of Fragile X hippocampus is that in this region, there's neural stem cells. They can become they can differentiate the neurons and integrate in neural circuits. And this has been found in almost all the animal studies so far, including humans. This neurogenesis process is important for learning, memory, anxiety, emotion, and also has been shown to be impaired in a number of disease conditions, including depression and aging neurodegeneration, as well as autism spectrum disorders. And intriguingly, this process can be enhanced by exercise and also by enriched environment. So when we started on this project, the question we want to ask is, in this hippocampal region, whether the FMRP is plays important roles in regulating this neurogenesis process, as demonstrated in the schematics here. In this region, there are neural stem cells in the blue-colored neural stems and progenitor cells. They can self-renewal. They can differentiate into immature neurons and mature neurons, and as well as astrocytes. So our question was whether FMRP plays important role in regulating these neural stem cells, and whether this regulation is important for cognitive function. This project was led at the time by Wei Xiangguo, a postdoc fellow in the lab. Now he is a professor at CAS Beijing. What we did is that we used a mouse model created by David Nielsen, which has a conditional flux FMRP, so we can delete it in Cre-expressing cells, and cross it with the nesting CRE-RT2 mice generated by Amelia Ash at the time. And so what we did is we deleted the FMRP only in neural stem cells and the subsequent progeny of neurons. And these mice exhibit reduced learning memory. And this is really interesting because the deletion of FMRP is only in the hippocampal neurons. And then we did another experiment utilizing another really creative line of mice generated by David Nielsen, which has a conditional on of FMR1. In this mice, the FMRP is expressed at very low level, lower than 10% of regular normal level FMRP. And we restored FMRP only in adult neural stem cells and new neurons. And we could restore at least partially the learning memory deficit. So this initial study really support our notion that FMRP plays important role in neural stem cells, and this regulation of FMRP is important for cognitive function. In order to understand how FMRP regulates neural stem cells, we dissected out the hippocampus and dendritic gyrus and dissociated cells and isolated neural progenitors in culture. And these cells in the culture condition, they can proliferate, and they express an immature marker, immature stem cell marker, nesting, and the SOX2 as shown here. And they can also be differentiated into neurons and glia in culture as shown here, and neuron in red, glia in green. So this system is a really nice in vitro culture system for studying neurodifferentiation and neurogenesis. So using this method, we assessed FMR1 knockout neural stem cells versus normal neural stem cells isolated from a mouse brain. So this is a data publishing our first paper showing that in the absence of FMRP, this is the knockout cells, these cells proliferate more and they differentiate into fewer neurons and more astrocytes. So this data actually we have repeated in many other studies presented in our subsequent papers as shown here. So because we have this really nice system with a nearly pure population neural stem cells in culture that can exhibit such a profound and consistent deficit in neurodifferentiation, we used this system as a model to study how FMRP regulates neurodifferentiation. For the past 15 years, we have published a number of papers and to use this model to study how FMRP regulates neurogenesis and neurodevelopment. And the results is summarized here because these are all published paper. I'll just summarize what we have found, we have discovered using this model. So we discovered that FMRP is actually put a brake or repression on a number of important signaling proteins, including GX3 beta, which is inhibitor of Wnt signaling, and MDM2, ubiquitin ligase. EP300 is a transcription factor, histone acetyltransferase. In the absence of FMRP, the brake is off, and these signaling molecules are overexpressing or overactivating. So what we show using mouse model is that we can actually inhibit these signaling pathways using small molecules, and these are all well-known small molecules and used by some of them are used by in clinical trials for other disease. And when we inhibit them in mouse models, and we could actually rescue both neurodifferentiation of neural stem cells and also the mouse behavior deficit present in the VEGF-X mutant mice. And one of them is currently under US patent. Also, we use this neural stem cell model to investigate novel mechanism how FMRP regulates transcription and translation. So this was a collaboration with Joe Richter at UMass and his postdoc Bao Liu at the time, and my postdoc at the time, Kevin Lee, who is now a professor at Tianjin University. So in this study, we used the neural stem cells exhibiting strong phenotype in proliferation differentiation to assess how FMRP regulated gene expression. So what we did is that we treated wild type and knockout neural stem cells with cyclohexamide to stop protein synthesis, and then we collect the cells and send it to Joe. And what he did is to digest this lyse it with RNAs to chop off the ribosome unbounded region, so we can obtain the footprint by RNA sequencing. He also sequenced the total RNAs comparison. So we discovered a large number of differentially expressed genes in several different manners. It's shown here. This is a summary of all the genes exhibiting differential expression in both mRNA level and the translation level, which is a ribosome footprint protected level. So as you can see, there are at least 6 different manners of gene expression changes, and we discovered here. And some of them only have translational changes. No matter how much the RNA is expressed, they either have an increased translation in the absence of FMRP or decreased translation. And others have a translation level did not really change by itself. It's all because of the level of mRNA change, which is along this diagonal, either up or down regulated changes. But there are also 2 groups of really interesting genes, because their mRNA level may actually be either upregulated or downregulated, but their translations go opposite direction. So we call them buffering down and buffering up. So, and when we when we try to understand what type of genes are going through this different type of regulation, we found something really also really interesting, as shown here in the gene ontology analysis for biological process and cellular components. As you can see here, the genes actually regulate transcription and also cell genesis, cell proliferation, and neural stem cells are mostly regulated at the mRNA level, either up and down. But the genes are actually regulating neural differentiation and synaptogenesis are mostly regulated at the level of a translation, the translation up or down. One very interesting group of genes actually is buffering down is the mitochondrial ribosome related genes. And this is the first time actually mitochondria appears directly in Fragile X literature in terms of changes in the context of absence of FMRP. So we did a quick analysis of the mitochondrial function using this seahorse assay. We put a neural stem cells from knockout mice and the wild-type mice onto the seahorse 96-well plates, and by adding different inhibitors of a mitochondrial respiratory chain, and we could actually assess the oxygen consumption rate of the cells, and we found that the knockout cells have a reduced oxygen consumption rate. So while we're doing this project, and we actually this discovery of mitochondria changes was actually hasn't been shown in literature at that time. So this really shed light on a separate project that we actually decided to move forward with, and this is actually a project actually we decided to study also in the context of neurogenesis in the hippocampus. This project was led by first led by Feifei Wang, who's now also professor at the Fudan University, and subsequently led by Mingjie Shen, together with a research intern, Michael Stockton. So what they did is, they first used hippocampal neural stem cells as a model to understand how FMRP regulate neural differentiation. And then they decided to zoom in onto this neuron maturation process in immature neurons. And the method that they used is to use retroviral targeting to specifically delete FMRP in the immature neurons. This is the strategy they used. They use retrovirus expressing Cre recombinase, but driven by the double coordinating promoter, which is the gene expressed in immature neurons. And they inject retrovirus into the hippocampal region of mice. As you can see in the wild-type mice, the retrovirus, because the retrovirus only targeting dividing cells, would actually, in fact, this type of stem cells, but the deletion only happened in immature neurons. And in the conditional knockout mice, when we deleted FMRP from immature neurons, the neurons develop into impaired dendritic morphology and shorter dendritic length. And also, interestingly, when they injected this retrovirus into the conditional on mice, which actually only restored FMRP in immature neurons, they could rescue the dendritic branching deficit, as shown in this black line. So this indicates that FMRP play an important role in not just the neural stem cells, but also in the immature neurons in terms of neurodevelopment. And this part of the quantification was done by a research intern at the time, Shan Kannan. Now he's a grad student in my lab. So the next, of course, because we have found FMRP's important function in immature neurons, our question is, what is the underlying mechanism of FMRP regulation of immature neurons? So to investigate this question, we use another mouse genetic model. We cross FMR1 knockout mice with double coordinate DS red mice. These mice express red fluorescent protein in only immature neurons. And we, using FACS, we dissected out this region of the cells, dissociated, put on the FACS sorting machine, and isolated this DS red high expressor cell population. You can see compared to negative controls. And then we use RNA-seq to look at the differential expression in the immature neurons and in the context of FMR knockout. And so we discovered that one of the top category of genes with exhibit significant changes is metabolic process genes. And as you remember, we discovered that mitochondrial deficits in the neural stem cells in Fragile X isolated from Fragile mice. So we decided to investigate the mitochondria in neurons. So this is a lot of data. I just presented one of the representing data. We use a primary neurons isolated from a wild type mice and from a knockout mice, and transfected neurons with GFP that fill all the neuron processes, or the mitochondria, actually together with the mitochondria targeting red fluorescent protein. And it's so that we could actually assess the mitochondria shape and the number. And we discovered that there's no differences in the overall mitochondria volume or area, but the mitochondria are rounder, with which is represented by this this quantitative measurement called aspect ratio. So this suggests that in FMR knockout neurons, mitochondria is more fragmented. So what does this mean? And in fact, actually, when we see this data, and we went back to literature, try to understand how mitochondria fragmentation can be a result of dysregulation. So mitochondria are always in this balance of fission and fusion, and this fission-fusion process actually is well controlled by a number of molecular pathways, and is really sensitive to the state of metabolism in neurons. And so there are a number of genes known to regulate fission, and including this well-known gene, phase 1, DRP1, and also a number of genes known to regulate fusion, including mitofusins 1 and 2 and OPO1. So the first thing we did is to look at the expression level of these genes in the FMR knockout neurons. What we found is that the fission genes, phase 1, DRP1, didn't show significant changes in the FMR knockout neurons, but the fusion genes, MFN1 and 2, both exhibited reduced levels. And the OPO1 didn't show significant change. So this suggests that the mitochondria in the knockout, FMR knockout neurons may have a fusion deficit, which leads to this fragmentation of mitochondria. So of course, we need to really use bioassay to validate there is indeed a fusion deficit in neurons. So we collaborated with my colleague Ed Chapman in my department and his postdoc Jason Viva. What we did is that we transfect neurons, mouse primary neurons, with this UV convertible fluorescent protein MEOS 3.2 mitochondria targeted, and the data is shown here. And so when we transfect neurons, all the neurons are green, but when we using UV only activate locally this fluorescent protein, they turn red. And then we use a live imaging to monitor the spread of the red fluorescence. If mitochondria are fusing to each other, the fluorescence will spread. If they don't fuse with each other, the fluorescence will stay put. So as you can see that in the knockout neurons, the spread of the fluorescence is a lot reduced. And here is the quantitative data to show that in the wild type neurons, the red fluorescence spread much faster, and which monitored at 5 minutes time point compared to knockout. So in this study, in summary, we discovered that FMRP is a regulator metafusing, particularly metafusing 2, which is important for mitochondrial fusion balance. And this is important for neuronal maturation, at least in the context of mouse model. And we also discovered one of the mediator of this regulation is the Huntington protein HTT. And we show that in the absence of FMRP, the metafusing level is downregulated, leading to mitochondrial fragmentation and impaired neural maturation. So while we identify this molecular mechanism, we always have this question whether we can use this knowledge to treat actually Fragile X mouse model. So Minjian and the leading scientists of this project decided to try this compound called M1, which has been shown in cultured cancer cells that can increase mitochondrial fusion. So he injected mice with M1 and then subject wild type and knockout mice to 3 different behavior tests. One is open field activity to assess the hyperactivity phenotype shown in F5X mouse model. And another one is a novel location test to assess the working memory, which has also been shown to be impaired in F5X mouse model. And the third test is a social interaction, the social novelty test, using 3-chamber test developed by Jackie Qualley. And here's the data. So the blue is vehicle treated and the red is M1 treated. As you can see that in the vehicle treated mice, knockout mice is hyperactive and also have impaired working memory and also have a reduced interest in novel stranger mice. And when we treated them with M1, and these deficits all seems rescued. So this, although the mouse model really present an opportunity for us to understand the mitochondrial function in the context of F5X mutation and present an opportunity for targeting mitochondrial deficit as a way of treating F5X syndrome. So as a summary of my first part of my talk, so we have used mouse neural stem cells, and we're still using these neural stem cells as a way to discover mechanism how FMRP regulates neural development and identify novel targets and molecular pathways mediating FMRP function. And in addition, in the more recent data, we found that mitochondrial deficit is likely major, if not only the contributor to neurodevelopmental alterations in F5X syndrome. So as you can see, and I love this cartoon that's actually I downloaded from a FRAXA website, and we tried very hard to use a mouse model to knock out the F5X syndrome problems. However, these are still mouse model studies. And in the past 11 years and 10 years since I moved to Wisconsin, I have tried to translate what we discovered into human. However, as you can see, there's a big difference in the size of brains between mouse and the human. And if you enlarge the mouse brain to the same size as human brains, they look very different. So the human brain is a lot more complex and with also with a much longer developmental process, and actually also have a different genetics. So in a recent publication, a review article written by me and Anita Bhattacharya, we actually listed all the things that are different between mouse and human, and why actually we must tackle this challenge to translate what we discovered in mouse into the human models. So one of the major differences between a mouse and a human in F5X syndrome is this genetic differences. So so many of you already know that in humans there are about 30 CG repeat at the 5' untranslation in the FMR1 gene. And in the F5X syndrome, when the CG repeat expanded mutationally to over 200, there's epigenetic demethylation and the gene shutdown of this gene expression. So although this is a mechanism in majority of the F5X syndrome in humans, we don't have any animal model that can recapitulate this mechanism, which makes it difficult to actually fully translate a mouse study or any other animal models into human patient knowledge. So fortunately, when I moved to Wisconsin, and Anita Bhattacharya, who's actually now a really close collaborator with me, and we've been working together for the past 10 years. And she already generated several induced pluripotent stem cell lines from F5X patient-derived skin cells. So these cells actually, they can propagate in culture. They also have a gene shutdown and demethylation similar to what's found in F5X syndrome. So using these cells, we have worked together trying to answer a number of challenging questions in the Fragile X field and trying our best to translate animal studies into human knowledge. The very first study we actually worked together on was also partially funded by FRAXA, is that we try to see if we can reactivate the silent FMR gene as a way to treat Fragile X syndrome. So as you know that in the Fragile X syndrome, the FMR gene is shut down. So how do we do this one? So as some of you know that people have been treating the Fragile X lymphocyte cells or even the iPS-derived cells. However, a readout has always been an issue. How do you actually quickly read out, efficiently read out FMR1 gene reactivation so that you can use it as effective tools for drug screen? So Meng Li was a postdoc in my lab, and now he's a professor in China. So he used gene editing, created this luciferase reporter line that put the nanoluciferase at the C-terminal of an endogenous FMR gene, so that the luciferase activity can represent the reactivation or expression of FMR gene. This line is published. We published in 2016, and now it's distributed by YCell. And you can, for anybody who is interested in using this line, you can actually order from YCell. And so I just show you one piece of data showing that when we treated with a 5-aza-deoxycytidine, which can partially demethylate the FMR1 gene promoter, and in dose response manner, we can see luciferase activation, which corresponds to the mRNA level of both nanoluciferase and FMR1. And of course, in high dosage in the human neural cells, this the 5-aza-deoxycytidine is toxic. More recently, we actually used this this system to screen drugs. This was done by an MD PhD student, Jack, who last year defended his PhD part of the thesis, and together with Ryan Risgaard, who was undergraduate student in my lab, and also he received a summer fellowship from National Fragile X Foundation to actually to carry out this study with Jack. They screened a large number of compounds, basically anything we could find. And in this is a summary of one of the largest studies, over 300,000 compounds. They identified over 200 potential positive hits, and eventually 2 likely positive hits. And this paper is now under review. So we hope to share with you in the near future. And so this is one study we've been carrying on for the past 10 years, and it has been challenging. But we along this time, this effort, we have created tools that other people can maybe use to further this investigation. The second question we try to answer using human stem cells is whether FMRP is important for human neurodevelopment. This study was carried out by Mingjie Shen together with research intern Tomer in my lab. And what Mingjie did is to utilize the human Fragile X iPS cells as a model to study in vivo neurodifferentiation. Because we do not have an animal model with a CGG repeat gene inactivation as shown in humans. So Mingjie differentiated the iPS cells into neuroprogenitors and labeled them using lentivirus expressing red or green fluorescence. And then he transplanted these cells in the neonatal cortex of mice. And these are skin mice, immunodeficient mice. And then after 4 months of to allow Fragile X or control human neuroprogenitor to differentiate and developing neurons in mouse models. And then he analyzed the neurodifferentiation. The reason we waited for 4 months is because the human iPS cells and neurodifferentiation follow the timeline of human development, which takes the gestation, as we know, it's 10 months, and the same thing as happened here, and really needed about 4 months for all the cells become neurons in vitro or in vivo. So here's the result. Mingjie analyzed the layer 4 human neurons localized in the layer 4 of mouse cortex and trace them using a neurolucida. And he found that in the absence, in the Fragile X human neurons differentiate into neurons with reduced dendritic complexity compared to the control iPS differentiated neurons. He also analyzed the mitochondria shape. They're also smaller and rounder. And also he found that these neurons also have increased oxidative stress level, similar to what we found in mouse models. So this is a still ongoing study, and Mingjie actually also received the FRAXA postdoc fellowship in the past 2 years to expand this study and into also drug screening of mitochondria activators as a potential way to identify modulators for human neurons. So this study we plan to publish in the next year or so. And another question that we try to answer using human neurons is how does FMRP regulate the human neuron development? So this is actually a very tricky question because there are many people trying to understand this. As summarized in this figure, which was published in a review article Joe Richter and I wrote for Nature Review Neuroscience last year, FMRP is known to have many different functions. And in addition to mRNA binding, it can also regulate transcription. And also FMRP has been shown to interact with a protein and regulate channels, ion channels directly. However, if you look at the FMRP function, most of its function, at least the well-established functions, are through interaction with binding of mRNAs. So we reckoned if we can identify FMRP bound mRNAs in human neurons, this will shed light on the potential mechanism how FMRP regulates human neurodevelopment. However, one of the challenge we have is we had and we still have is that antibody against FMRP is not really good for either IP or protein interaction analysis. So what do we do then? So what we did is Meng Li in my lab again, and he used gene editing, added a flag tag, 3X flag tag at the C terminal of the endogenous FMR1 gene, so that the flag tag's expression, the FMR1 flag fusion protein is expressed under endogenous from the endogenous FMR locus. So this gives us a way to monitor FMRP protein expression and also identify FMRP interacting mRNA or protein through endogenous level. So what he did is to differentiate the human pluripotent stem cells with endogenous FMR flag FMRP expression into neuroepithelial progenitors and then into the lineage of dorsal excitatory neurons or the lineage of ventral inhibitory neurons. So because there's a step of dorsal NPCs, we also analyzed the neuroprogenitors that's on the way to become a dorsal excitatory neuron and the ventral progenitor on the way to become ventral inhibitor neuron. So here's a histological assessment to show that the dorsal, the D neurons do express the glutamate, vascular glutamate transporter, which is a marker for the excitatory neuron, and the ventral neuron express GABA, and they all express a flag tag. So these cells provide a model for us to investigate what mRNAs are bound by FMRP. And we use a method called CLIP, cross-linking IP coupled to next generation sequencing. So what we did is we cross-link FMRP, which in these cells are flag-tagged FMRP, with mRNAs they bound to in the cells. And then we use antibody beads that's against the flag tag to pull down FMRP together with the mRNA they bound in these cells. And then we isolate RNA and subject the RNA to next generation sequencing. And here's an example of the FMRP targeted or bound mRNA in human dorsal NPCs and the human dorsal neurons. As you can see, some of the well-known clusters involve neurogenesis, development, signal transduction, small GTP as clusters are shown here. So when we have this data from 4 different cell types, dorsal, ventral, NPC, and neurons, we compared our data to the published mouse CLIP-6 data published by Jen Darniel's and Bob Darniel's group in 2011 in the Cell paper, and more recently by Maureen et al. In 2018. And we only compare neurons because they're more relevant to this to the mouse forebrain CLIPS data. As you can see, there are actually shared targets among all different, both human neurons and the mouse brains. But there are also unique targets that only in human neurons and only in human dorsal neurons and the ventral neurons. We also compared our identified FMRP targets with the disease GeneText database. And we discovered that many of the targets are involved in the human intellectual disability and Fragile X-related symptoms. In order to understand how FMRP regulates human neural differentiation in neurogenesis, we also created a FMRP-deleted isogenic human pluripotent stem cells by inserting a stop codon in the 3rd exon. And here's a Western blot showing the absence of FMRP expression in the 3 different lines: the parental line H1, H13, ESL lines, and GM1 is iPSL lines, and convert to wild type. And then we did RNA-seq transcriptomic analysis of NPCs and dorsal ventral NPCs and dorsal ventral neurons. And here's one example showing the differential expression, both up and down regulated genes in the absence of FMRP. So one of the observations we had, which was puzzling at the time, is we discovered that there are very few genes overlap between FMRP bound, which is CLIP-6 data, and the differentially expressed genes, which are RNA-seq data. So we realized that we should really integrate these 2 sets of data to understand how FMRP regulates neural stem cells and neural development. So we interact and collaborate with Dr. Sushmita Roy, and she's bioinformatics faculty at the University of Wisconsin, and her postdoc Junha Xin. As you can see here, that they used integrated network analysis to integrate both CLIP-6 data and RNA-seq data in 4 different cell types. And as you can see, overall, there are similar clusters among all cell types, but there are differences in the amount of genes and the size of the clusters. We discover the significant differences between NPCs and who are proliferating and neurons that's post-mitotic. For example, this cluster is present in NPCs but absent in neurons. And we also discovered that dorsal ventral neurons have a different set of networks regulated by FMRP. For example, this cluster in ventral neuron, which is not in the dorsal neuron. So one of the knowledge we gained from this analysis is to understand how FMRP regulated regulate genes at both through direct targeting a set of genes and indirectly regulate transcription. As shown in this particular network, FMRP binds CTNNB1, which is coded for beta-catenin, one of the Wnt signaling mediator. So beta-catenin, CTNNB1, it's derived bound by FMRP, but its transcription is not changed. The mRNA level is not changed in the knockout FMRP neurons. However, you can see all these genes with the differential transcription level. These are present in the knockout cells, in FMRP knockout cells. They are not the direct target of FMRP, but they are regulated by this beta-catenin. So this really presented an interesting scenario is that FMRP can actually directly regulate a set of a protein or mRNA, but indirectly regulate many other genes. So really kind of substantiate this notion that FMRP is really have many different functions that, especially in different cell types. Finally, I want to present that we currently try to tackle this new challenge. It's actually not a new challenge, it's an old challenge, is that among Fragile X individuals, there's a large phenotypic diversity. As many of the physicians and the family know that not all the Fragile X patients are exhibit similar kind of phenotypes. And this actually was quantitatively identified by Dr. Craig Harrison using a non-invasive EEG method. So in this paper published from his group in 2017, he showed that when he used the resting gamma EEG as a way to assess the Fragile X patient and compared to controls, he found that there's a large spread in the resting gamma power among Fragile X patients, especially among the male patients. And a small portion, about 1/3 of patients, exhibit high resting gamma and low alpha power, and while the other ones are more comparable with the controls. So this is only one of the heterogeneity among Fragile X populations. And Lisbeth Kravitz, who's another physician scientist, also discovered a large heterogeneity among Fragile patients in terms of their cognitive functions and their behavior. So one of the intriguing discovery made by Craig is that the patients exhibit different EEG high versus low gamma powers also respond to several clinical trial drugs differently. So this implies that so we may be able to differentiate this the patient population through EEG as a non-invasive way to stratify patients. However, if we want to test clinical trial drugs or for preclinical drug development, so it is challenging to always involve patient population and EEG in all the studies. Can we actually use iPS cells as a model to carry out the initial preclinical analysis or even clinical trial drug testing so that we do not have to always go to the patient, so that maybe we can differentiate the patient who may have a differential response to drugs. So this is the question we had for several years. Finally, we were able to start working on that. So this is a close collaboration with Craig Harrison and together with Anita Bhattacharya in my center. And this study is led by a postdoc fellow, Carissa Saroyas, who recently was awarded the postdoc fellowship from FRAXA to really push forward this study. The overview of the study is shown here, created by a figure created by Carissa. So Craig Harrison, the physician scientist who many of you are really familiar with, and has identified patients with a high gamma and low gamma, and also as well as matched controls. And with funding from a John Merck fund, we were able to derive iPS cells from these patients. And totally, we have 17 lines from 17 individuals, with 11 of them from the Fragile X with high or low gamma, and the rest from controls. And we were able to actually characterize these iPS cells and also deposit them into white cells. So upon publication of our paper, these cells will be available to the scientific community. And with the recent funding from FRAXA, we are aiming to differentiate neurons and using multi-electrode array to assess their hyperexcitability, their differences in the and also different differential network activations in culture. And in the meantime, we'll carry out transcriptomic analysis to understand the molecular underpinning between this high gamma, low gamma EEG among Fragile X individuals. So this is an overlay of this project. I want to just show you one piece of data, preliminary data that we had when we were able to obtain using John Merck funding. And here's showing that the weighted firing rate is the black is the control and the red and pink are Fragile X. As you can see, over several weeks of recording, both Fragile X lines exhibit hyperactivity over time. However, when we assess some other parameters, for example, this one is to measure the spike regularity within network bursts. The two Fragile X lines are quite different. And this is a preliminary data. We are analyzing all the trails, the high G, low G, and the controls we have in hand right now. And hopefully in a year, we will be able to report to you that we have discovered. And our goal is if we can identify the MEA signature between high gamma, low gamma patient lines, then we can use this method maybe to assess some of the clinical trial drugs or potential compounds that may exhibit differential impact on diverse Fragile X patient populations. So with that, I'm going to summarize my talk. So in my talk, I first talk about using mouse neural stem cell models as a way to understand the Fragile X mechanism of pathogenesis and functional FMRP. And in second part of my talk, I presented what we are doing using human stem cell models. We try to identify compounds that can reactivate, silence the gene, and we investigate functional FMRP in human development. And we try to our best to identify FMRP targets in the pathways in human neural cells. And currently, we actively pursuing the possibility of using human iPS cells as a model to understand and investigate diversity among Fragile X patients and the drug responses. So with that, I'm going to thank. I actually have a lot of people I need to thank. First, I want to actually thank my lab members. These are the current lab members, and the one with yellow are those who contributed to the study I presented today, and also some past lab members. And I also want to thank the collaborators. Many of them I actually met and interacted during FRAXA meetings and or Gordon conference, partially hosted by FRAXA. And in addition to that, I would like to thank the funding agencies who actually provided us with means to investigate these questions. And I want to say that although the FRAXA grant has always been at the initial stage of each of these projects, these are really important seed money for us to explore novel questions, and without this kind of funding from FRAXA, from John Merck, we will not be able to actually to conduct some of the more exploratory and high risk, high reward type of investigation. So thank you very much. Thank FRAXA, and thank all the Fragile X families who support FRAXA, who support Jenny Kyle Fund, support John Merck, Autism Speaks, and National Fragile X Foundation, also Brain Research Foundation. Even though these are all private foundations, they are just as important as the NIH for promoting biomedical research and for understanding human disease. And know for the, I'm talking about Fragile X, but many of the knowledge and the methodology we developed can also benefit many other disease, not just neurodevelopmental disorders. So I want to thank you, and I think that's the end of my talk.
Thank you very much, Xinyu. That was fantastic. I know we've had a large number of participants on today's call, and I'm sure we'll have plenty of questions. We also have plenty of time for questions. And the way you ask a question is through the chat function on Zoom. So we have a few in the queue already. And we'll be getting started on those momentarily. But if you have a question, just put it in line through the chat function. All righty, so and I'll just read these into the record, as it were, for our recording. From a user via direct message, I'm wondering, when could a cure for Fragile X using CRISPR Gold be available? CRISPR Gold was a news item a while back, and you've mentioned, Xinyu, CRISPR as a research tool, and a very important research tool. What about CRISPR as a therapeutic?
That's a good question. I know several labs proposed to do that, trying to use CRISPR as a way to maybe delete the long CG repeat. And we actually also tried in my lab. It's actually because of this long CG repeat is also epigenetically silenced the repeat. It's really difficult to delete specifically from the Fragile X gene. We have not been successful, and it would be great to see if somebody can do that. But so far, all the published papers have not shown high efficiency in terms of a specific deletion of the CG repeat. So, yeah, I think that's a good idea.
From the layperson standpoint, the takeaway message I've gotten from a number of studies, and Steve Warren did some early work on this, is that you not only need to cut down the repeats, but you need to still reactivate the gene. So it really is a two-step process. And it's a little bit more complicated than we might like, but that's just the way it is. A question from Judy Kelleher. Hi, Judy. Related to the original neurogenesis differences in hippocampus, did you ever look at the SVZ stem cell niche?
We actually, for that study, we didn't look at the SVZ, and to be honest, at the time, we kind of focused on one region, and when we wanted to look at the other region, there was a controversy whether SVZ neurogenesis is actually relevant in adult. So we didn't, but some other people have a look at that. And so, and I actually don't remember exactly what they found. I'm sorry, it's been a while. So, yeah.
From Jean Ascaria, I was wondering about the buffered mRNAs. Is there any structural component in these messenger RNAs that could explain why the levels are changing while the translation is unchanged? Do they bind certain RBPs other than presumably FMRP? Are the mitochondrial ribosomal mRNAs buffered?
That's a very good question. Actually, we actually didn't know. So I know Joe has been working together with Kim Huber on the ribosome footprinting in neurons and identified the subset of genes really regulated by FMRP in terms of the translation efficiency. However, for the buffering group, we actually didn't really pursue that. And just recently, we have some preliminary data showing that it is possible that some of this buffering is because of protein cofactors or protein target of FMRP may be ubiquitinated, so that we could actually have this increased transcription, but a downregulation of translation, vice versa. But we don't really have a firm data to support that yet. So it's a good question. Actually, now I really need to work on these questions. So
this happens in every presentation, that whenever there's one finding, you can go off in any one of 20 different directions, and you can't chase down every lead. So in fairness to the speaker, you can't pursue all of these leads.
But I love the question, because I think sometimes we kind of publish a paper and move on to the next thing that we're really interested in, and then when giving presentation of this data later, when people ask questions, we realize, oh, there's things we could do too. So maybe I can go back to that to answer these questions.
Why meetings are important. From Tatsuaki Kurosaki, based on your criteria, mitochondrial ribosomal genes are generally buffered down in FMR1 KO mice. Is FMRP directly involved in the translation of these transcripts? If so, FMRP functions as a translational activator instead of a repressor.
That's a very good question. I think I remember the study showing that FMRP may be translational activator. And we have not looked into that yet. And again, so this is a really nice question. And together with the last question, really now motivate me to go back to that data and work more on that system. So thank you very much. I'm sorry I can't really answer a question right now.
We'll try this other one from Tatsuaki. He says your FMRP ClipSeq detects more FMRP targets in neural progenitor cells relative to mature neurons. Do you know the expression of FMRP, mRNA, and protein if it's changed during neurodevelopment from NPCs to neurons?
So we know that FMRP is expressed at higher level in neurons than NPCs. However, one of the reason maybe ClipSeq we see more targets in NPCs is because we can actually get a lot more NPCs than neurons. One of the major challenges in doing ClipSeq is the cell number. And because of NPCs, we can get a lot more number. The IP efficiency is better. That could be one of the main reasons why we get more targets. And the neurons, actually, when we differentiate human NPC into neurons, we have to actually differentiate them in low density. Otherwise, the percentage of neuron would reduce. We didn't really want to use a direct differentiation because we didn't want to bypass the NPC stage. We want to really model the different human kind of in vivo differentiation. So the number of neurons we obtained was much smaller. And that's why actually most people don't use human NPC or neurons. They use like a HEK cells or HeLa cells to do this kind of a clip analysis. So your questions are really good. And we do need to go back. If we can actually increase the efficiency of ClipSeq, we may be able to actually repeat this one at a higher cell number. But at this time, we indeed obtain more targets in NPCs and neurons.
Question from Rocky Roy. Interesting talk. From the preliminary data presented on 3 lines, does the multi-electrode array, or excuse me, microelectrode array firing pattern data correlate with resting gamma EEG measurements from patients?
That is our project right now. So we were able to derive the iPSCs with the pilot funding, and we analyzed one set of trio, the high and low gamma and the control. That's what we see. So like we're currently differentiating additional trios. We call trios because there's 3 at the time we differentiate, and to see if we can identify unique or specific or consistent signature of in MEA that correct corresponding to the EEG. And then we've been talking to Craig Arison because he's the director of the new FRAXA Center at one of the 3 centers funded, and their focus is on using EEG to study in FRAXA both mouse model and the human. And so they have been developing method for data analysis. And our goal is to correlate our data with their data analysis to see if there's any consistent signature that we can use in vitro to represent the patient EEG changes in vivo. So yeah, that's our project right now. So.
And from Kagistia, who's, if I'm not mistaken, a FRAXA fellow in Singapore. So talk about global reach. Fantastic work, Dr. Zhao. Do you perform the reactivation screen in the neural cells derived from the nano-loose Fragile X cell iPSCs, or were they done in iPSCs? For the MEA-based readout, how do you differentiate the firing rate patterns as a majority of Fragile X neurons are hyperexcitable?
So for the first question, so the nano-look was that line was created in iPS cells, Fragile X iPS cells with a patient. It's a patient-derived iPS cells with a long CG repeat. There's no FMR1 expression. And our screen was done on NPCs because we're using this 1536 really well plates. You actually have to be able to expand it. And if you use neurons, it will be really, really hard to do large scale screens. So we use NPC, neural progenitor cells, for the screen. And so that line is available. I know several groups are already ordered from YCell and carry out experiments in their own group. And I highly encourage you to try it too if you're interested in this topic. And in terms of MEA, and MEA can measure basically the overall activity of culture cells. It doesn't really discriminate where it measures. So it's not really the most precise way of measuring neuronal activity, but it's a good, almost high throughput result. It gives us a way to look at the hyperexcitability of neurons and also network firing, the synchronized activity of neuron and dish. So our plan is using MEA, the multi-well MEA, 96 wells or even more wells, to do the initial screen. And then we use a more classic way of calcium imaging patch clamp to validate the finding. In terms of using this system as a way for drug screen, the patch clamp and it's just too low throughput. Calcium imaging also right now is not really high throughput either. So MEA provides a possible way for relatively high throughput screening of drug response. So we think the MEA is valuable in that aspect.
If I could just ask a little follow-up question. I believe the preliminary data that you presented on that first triplet showed that in terms of just the flat-out firing rates, the Fragile X cells, both the high gamma and low gamma, were both considerably higher than the control cells. Yes. Whereas it was more of an internal parameter of the firing rate that differentiated the high gamma from the low gamma Fragile X cells.
Yes. So we actually found that there are a lot of shared characteristics between the two Fragile X line compared to control, but the two Fragile X line indeed exhibit differential characteristics. Because this is only from one trail, we want to make sure that we identify, in the next 5 trails, we can identify signatures that we can say, oh yeah, these are the signature for high gamma, this is low. And when we treat with some of the compounds that, Craig Arison already showed, some compounds have a differential response from Fragile X patients with high gamma versus low gamma. We want to test them to see if we can also see differential response in culture neurons. So hopefully we can identify something where we can say, okay, these are the signature we can use to differentiate these two groups. So that's our ultimate goal. So, yeah.
Question from Maya Castron in Finland. Very nice talk. Does high gamma power correlate with clinical epilepsy phenotype?
Actually, I don't remember that's one of the phenotypes shown by Craig, because we actually look at the behavior phenotypes, and so definitely the patient with high gamma has a little more severe behavior and cognitive impairment, but I don't remember epilepsy is one of those impairments.
Yeah, I'm not certain what Craig's actual data show, and I guess we'll leave that to him to comment. Yeah, if he's with us today. I'm not sure, but it is in general. In general, there's a correlation between high gamma and more severe phenotypes, including autism and including seizures. But I'm not sure that that's what his data set shows.
I don't remember it's a seizure is listed on that, because I remember looking at the behavior list of aspects.
Could be too small a number to really show at this point. From Justin Fallon, as you know, there are promising results in Fragile X trials targeting cyclic AMP. Do any of your pathways intersect with cyclic AMP function?
So far, we have not really particularly looking at that cyclic AMP function, but in the integrated network analysis, there are cyclic AMP related pathway are shown in those networks. I have to go back to look specifically. And actually, the network really jump out is actually the AKT pathway and the PI3 kinase pathway actually are shown in those networks. I remember actually, if you remember Gary Basel's work that he looked at this pathway, and I didn't pay attention to the cyclin MP particularly.
Ilaria Bertocchi asks, the gamma differences also present in preclinical models. Thanks for the talk. And this, I guess, gets back to Devin Binder's previous symposium. So maybe that's a different talk where we're doing animal electrophysiology. And if I can recall his presentation, the answer is yes. We do see the high gamma in the preclinical model. That's using more of the other MEA, the multi-electrode arrays, as opposed to the micro-electrode arrays.
Yeah. So we have been, this project is actually started by a collaboration with Craig Arison group at the Cincinnati, and we still have the ongoing collaboration. Our goal is that once we have this IPS knowledge, we'll actually work with the Craig Center to see if we can compare directly our data versus their in vivo mouse and human EEG data. So it's really exciting. We don't know what exactly we're going to find yet, and we have some dreams and anticipations, and hopefully we would be able to share with you our discovery in the near future.
Question from Bharat Reddy. Great talk, Dr. Zhao. Have you looked into any glial cell roles in Fragile X?
Actually, we did a little bit several years ago using, actually, that's one of the graduate project, and we did find some differences, but we have not really repeated in terms of FMRP function in glia. And currently, I know there are two groups. Both we actually kind of work together and collaborate. One is they are working on the glial function Fragile X. One is Yungjie Yang's group at the Tufts, and the other one is on Dunaski at what's Kansas? No, Nebraska. Yeah. So they both working on the glial function of Fragile X. I'm still really interested in that glial genesis in Fragile X because we did see some differences in one pair of Fragile X versus control. However, glial differentiation in human cells take 5 to 9 months, and so it's a little hard to convince actually a student or postdoc to actually do one experiment that would actually need to wait for 5 to 9 months to do analysis. So we have not done extensive analysis in glial ourselves.
If I could ask a question from the moderator. Early on in your presentation, you showed and briefly mentioned the effects of curcumin. I happen to know that there are a number of people in today's symposium who are very interested in curcumin, including me. Could you say a little bit about what you've done with curcumin and why, and what kinds of results you've gotten?
Yeah, so the reason we use that is because we actually we were looking at FMRP targets in neural stem cells, and we published one paper showing that FMRP regulates MDM2 and the P53 in stem cell proliferation fit specification that in 2016. And then we want to look at the more like how FMRP media, because in that model, in the mouse model, we see that in young adult mice, the neural stem cells are overactivating, they are over-proliferating. And if we actually can treat them with Newton 3, we can even out that process. Then we say, okay, because it has been shown overactivating on your stem cells leading to depletion of your stem cells in adults. And that's actually is one of the key regulation of adult stem cells is maintenance of a stem cell pool for cell genesis and repair. So we look at the older mice at 6 months old, VIGX mice, we found that the neural stem cells actually are much lower. They don't proliferate much. They go almost depleted. And so we identify one of the targets actually is EP300 is an FMRP target in older neural stem cells, but it's not in the younger neural stem cells. So we try to actually activate it. So to inhibit the EP300 activity, one of the known inhibitor is actually this curcumin, and it's not really clean inhibitor. It does a lot of other things, but indeed has activity in inhibition of EP300 activity. So we did that, and we treated mice with curcumin, and we found that actually it can rescue the behavior deficit and also neurogenesis deficit in the FMRN mutant mice. These are 6 months old mice, considered as like a mature adult age in FIGX, in mouse models. So curcumin has been used for like, I think at least 100 clinical trials, different kinds of clinical trials. I don't know if all of these clinical trials have been published, but for many diseases, and it's over-the-counter supplement. So I would be very interested to know if curcumin indeed have a function in relieving VIGX symptom. So I'm not a physician treating patients, so I can't say, okay, do it. But it'd be great to see if someone's interested in testing the curcumin effect.
Oh, it is something that we're very much interested in. And I'm just wondering, how did you administer it to the mice in your studies?
Oh, so when we administer to the mice, we actually did IP injection because we always get a more consistent dosage when we do IP. And I know people have done like providing the drinking water or other means, but then that you really depend on how much they drink, and especially you can. We try not to house mice in a single cage because that actually those lonely mice, they don't behave very normally. So when you have 2 mice there, you have to figure out how much they each drink. So we found that IP injection will be the most consistent way. So we did IP injection. So
well, we are interested, and so I might be talking to you more about that. And of note, there are a lot of enhanced bioavailability formulations of curcumin that are available on the market now. That's one of the big problems that we've always had in using it off-label or doing clinical trials, is that you can basically take as much as you can possibly stand, and hardly any of it gets in. But now there are versions of curcumin that have 1,000 times greater absorption.
Yeah, that'd be great actually if someone can try, because we actually, when we found that, we're really excited about that, because the scientists, the postdoc scientists who actually wanted to use curcumin, because we have another compound. It's more specific for EP300 inhibition. It's called the C646, I think. But he said that compound is not a clinical trial compound, and also it's a chemical. So curcumin is, even though it's not as clean, it's over the counter. It's a non-invasive, non-toxic. So, and because he has background in Chinese medicine, he got his I think a PhD, bachelor degree from the Chinese medicine. So he wanted to actually try a compound that's not really chemical, but more people are more acceptable to use. And it actually surprised me. It really worked. So, but because it's over the counter and it's not something that it's patentable or anything, people may not even consider it as seriously as a potential way of a treatment. So it'd be great. I'm actually very interested in anybody who's going to try that. And
well, that's great. And Maria Modi asks, how long do these rescue effects last after administration? I'm not sure if she's talking about perhaps a carryover effect if you give the drug. With curcumin, for example, if you give the drug, or perhaps just more generally, any of these treatments of proliferation or treatments of
so that's a very good question, because we actually had this. Yes, so actually we only did one of them for long term. It's a Newton 3. After we published a paper in 2016 and showing Newton 3 can low dosage Newton 3 can rescue the behavior deficit. At the time, we also have some mice we injected, but because we didn't need them for the paper, we kind of let them go for 4 months, and then we said, why don't we test them? And my hypothesis is that the rescue is transient. After rescue, for the first month, you have to keep injecting them. But surprisingly, the rescue effect still stays at 4 months later. So we are actually right now analyzing these mice, and both in terms of neurogenesis and also the other brain regions, the total in the cortex, in the hippocampus, what exactly changed, after the injection. And so because this is 4 months after post Newton 3 injection, it shouldn't have a long-lasting effect like that, but it did. So yeah, so I wouldn't say that all of the treatment will have long-lasting effect, but at least in this case, our hypothesis is that there might be some epigenetic changes occurred, at initial injection, because Newton 3 affect MDM2, which actually downstream affect several epigenetic factors like HDX and P53, and which may have long-lasting effect. This is a project that we are trying to finish this summer, and by a grad student in my lab. So hopefully we'll have the answer for at least one of the treatment.
Fascinating. And so perhaps, just broadly speaking, when you're targeting neurogenesis, you can have some very long-lasting effects from relatively short-term treatment.
Could be. Those cells are really plastic. They are very responsive to an external environment, and so it could be actually the one of the mediator for long-lasting effect. So,
and if you would, another follow-up question that I have about the mitochondria. What are the metabolic signaling inputs into metabolic fusion versus fission and maintaining that balance in mitochondrial function? For example, there's been a lot of interest in insulin signaling in Fragile X and other metabolic effects that have been only more recently recognized. How do those hormonal influences affect mitochondrial function, or is that known?
Actually, if you read the review articles on mitochondrial regulation, there's tons of ways can affect mitochondria. And so at this moment, we actually don't really know how exactly FMRP-deficiency may affect mitochondrial function. We have some specific targets, like for example, we know that FMRP can regulate mitofusing, but not directly. We identify that HTT as one of the target that could be affecting as a mediator, but there are many other mediators. Many of the FMRP targets can actually directly or indirectly affect mitochondria. So mitochondria almost like this a sensor, kind of sensing the metabolic state, sensing of the neuronal state or activation state. So the question you ask is, I think, is a multi-billion-dollar question here. So, but no matter what, the mitochondrial deficit is a very consistent defect. That's why in the data I showed that on the Fragile X patient-derived neurons, high G, low G, both neurons actually exhibit mitochondrial deficit. So, and in also our existing iPS cell-derived neuron, we see mitochondrial deficit. So we are interested in, if we cannot find the root, maybe we can cure the phenomenon. We testing multiple mitochondria enhancing chemicals to see if we can actually rescue at the end of it. And luckily, there are quite a few compounds out there developed for many other diseases. Since mitochondrial deficit is really prevalent in neurodegeneration, with all this interest in Alzheimer's disease, there are multiple drugs are actually being considered now. So yeah, so I can't really answer the question. I really don't know the answer for what is link between FMRP and the mitochondrial deficit.
Good
But
question.
at least we can actually use the phenomenon as a way to see you can treat this deficit.
Well, thanks. We got one last comment. We recently performed a knockout in cell lines and found a lot of differentially expressed genes that have no direct link to the gene that is knocked out. And even when we knock out the same gene in different samples of the same cell type, we get different differentially expressed genes. So it appears we have a lot of unexplained stochastic effects. Could this have contributed to the variability in patients for IPS models?
So this is a good question, because actually I think of FMRP have different function in different cell types, depending on what cell type you're actually doing knockout. Or as a CLIP target analysis, you will get different results. So there's two publications from the Darnielle group, one on the bioRxiv, one I think is eLife. They did a cell type specific flag tag of FMRP and did a CLIP-seq. And they found a different set of genes are bound by FMRP and exhibit differential expression in cerebellum, in the CA excited neurons, and they have a different set of genes regulated by FMRP. So really, depending on what kind of cell types you're working with, you may actually have you discover different set of molecular pathways. There are some conversions, but there really are differences also. This is also the other kind of challenge in working with this protein that's expressed in many different cell types and many different types of neurons or glia. And when we try to treat the disease, we actually have to think about the impact on other cell types too. So I think it's important to understand cell type specific function of FMRP. So at least you have the knowledge of if you actually when FMRP absent from one cell type, here's your target, but what happened to the other cell type? So these basic studies are important in this aspect.
Well, this has been a fascinating discussion, and it was an outstanding presentation. And I just want to thank everybody who's attended, but most of all, I want to thank Dr. Xinyu Zhao. And we wish you all the best in your ongoing studies. It's really fascinating work. Thank you.
Thank you. Thank you, everyone. It has been really fun. So.
Thanks a lot. Bye-bye.
Bye-bye.